Good morning, students. Just continuation of the last lecture, roles of animals in medicine production. So, roles of animals in medicine production, and there are so many research and development goes on medicine production about the roles of animals in production of medicines, in science and in the area of medicines and all, pharmacy and all. And research and development, animals are used in preclinical trials to assess the safety and efficiency of new drugs. That is a role of animals in production of medicines for the purpose of research and development. Animals have been used in clinical trials, preclinical trials, and to assess the safety and efficiency of the new drugs based on animals, the medicines based on animals. So roles of animals in production of medicine or in medical production, you can say. Production of biologics, certain animals produce insulin, vaccines and antibodies. So production of biologics, that is a role of animals, importance of animals in production of medicine. Certain animals produce biological products like insulin, vaccines and antibodies. Then come to types of animals used. Small animals, mice, rats, rabbits are commonly used for laboratory research. Large animals, pigs, sheep, cows for larger scale biological production. So we have used different types of animals for the production of medicines in the research lab, in the laboratory research lab. We have used small animals like mice, rats and rabbits, and large animals like pigs, sheep and cows for larger scale biological production. And there is specialized use of animals, horses for antivenoms, dogs and non-human primates for certain disease studies. Examples of animal-derived medicines are insulin, historically derived from pig and cow pancreas, now mostly produced by recombinant DNA technology. Vaccines are chicken eggs, for the examples for the vaccines, for the preparations of vaccines from animals. Chicken eggs are used to grow viruses for flu vaccines. So we have used chicken eggs, right, from uh, chicken eggs we have used to grow viruses for flu vaccine. So we have made medicines. We have made medicines from chicken eggs. You should know these things. As a student, should know these things. We have made medicines from chicken eggs to grow viruses for flu vaccines, and we have used pig and cow pancreas for development of insulin DNA technology, and monoclonal antibodies, mice genetically modified to produce antibodies for diseases like cancer. Then alternatives to animal use. In vitro method, cell cultures and tissue engineering is there, and computer model simulations and bioinformatics is there. That is the alternative use of animal use. In vitro method is here. That is alternative to animal use. That is cell culture and tissue engineering. Computer models are there, simulations are there, bioinformatics is there, and micro dosing in humans, administering subtherapeutic doses to humans to gather preliminary data. Then come to animal-based medicines. Insulins are there, heparin is there. Insulin source is historically derived from the pancreas of pigs and cows, and use treatment of diabetes by regulating blood sugar levels. Development is now mostly produced using recombinant DNA technology, reducing reliance on animal sources. So these are the sources of insulin-based, animal-based medicines are used for the insulin purposes, for the insulin disorder and all, and for the treatment of diabetes and all, by regulating your blood sugar level and source is derived from pancreas of pigs and cows, and development now mostly produced using recombinant technology, reducing reliance on animal sources. This is a new development in the area of animal-based medicines. Now we are using DNA technology, and we have reduced a reliance on animal-based sources, medicines. That is a new invention, innovation in the field of medicines and pharmacy for the treatment of diabetes and all, for the regulation of your blood sugar level in your body. And earlier we have used pancreas of pigs and all, but now we have used there certain developments in the insulin medicines and all. Now we have used DNA-based technologies. And the next is heparin. Heparin is source extracted from the intestinal mucosa of pigs. That is the source of heparin medicine. And use is for anticoagulant, used to prevent blood clots and doing surgeries and in treating thrombosis. And production is pig intestine remains a primary source due to high yield and efficiency. That is the production of heparin. Pig intestine remains the primary source due to high yield and efficiency. Then antivenom is their source is produced by injecting horses with small doses of venom and then harvesting the antibodies developed in their blood. Use is treatment for bites from venomous snakes, spiders, and other venomous animals. Process is the antibodies, immunoglobulins are purified from the horse blood to create the antivenom. That is a source produced by injecting horses with small doses of venom and then harvesting the antibodies developed in their blood. Harvesting the antibodies in their blood, and then use that treatment for bites from venomous snakes, spiders, and other venomous animals. Process is antibodies are purified from the horse blood to create the antivenom. This is a medicine used, animal-based medicine. Vaccines are source chicken eggs are commonly used to cultivate viruses for vaccine. For use in influenza vaccines, among others, are produced using this method. Processes are fertilized chicken eggs, providing ideal environment for virus propagation, which is then harvested and purified for vaccine production. Then come to monoclonal antibodies, and these are the animal-based medicines. Monoclonal antibodies sources are mice are genetically engineered to produce human-like antibodies. Use treatment of cancers, autoimmune disease, and other chronic conditions. Development hybridoma technology involves fusing mouse cells with human cells to produce monoclonal antibodies. That is, a monoclonal antibodies. Mice are genetically engineered to produce human-like antibodies. Treatment of cancers, autoimmune disorders, disease, and other chronic conditions. Development is hybridoma technology involving fusing mouse cells with human cells to produce monoclonal antibodies. Hormones are their source. Pregnant mare urine is used to produce conjugated estrogens, and use is hormone replacement therapy for menopausal systems. Products pre-marine is well known HRT derived from this source, that is the hormones. A pregnant mare urine is used to produce conjugated estrogens, and who uses hormone replacement therapy for menopausal system, and products are pre-marine is well known HRT derived from this source. Then come to enzymes. Enzymes are the source is animal organs for making of enzyme medicines. The source is animal organs, particularly pancreas of pigs and cows, and use is for digesting enzyme supplements for individuals with pancreatic insufficiency. Products are pancreatin and other enzyme supplements. Then collagen is their source is extracted from bovine or porcine tissues. Use cosmetic treatments, wound healings, and reconstructive surgeries. Forms are injectable fillers, wound dressings, and scaffolds for tissue engineering. So these are the source of animal organs for enzyme medicines, particularly pancreas of pigs and cows. This is used for digestive enzyme supplements for individuals with pancreatic insufficiency or disorder, And products are pancreatin and other enzyme supplements you can give to the patient. And for collagen medicine, source is extracted from bovine and porcine tissues, and use is cosmetic treatments, wound healing, and reconstructive surgeries. Forms are injectable fillers, wound dressings, and scaffolds for tissue engineering. Then pepsin, source is stomach lining of pigs, and for pepsin medicine for the use of patients and all, and uses digestive aid in and pharmaceutical preparations for protein digestion. Application is used in digestive supplements and certain diagnostic tests. Thyroid extract is there. Source is glands of pigs and cows, and use is treatment of hypothyroidism. And products is natural desiccated thyroid medications like Armour thyroid. References are National Institute of Health or Family, Animal Derived Medicines, World Health Organization WHO reports on vaccine production, and peer-reviewed articles on development and use of animal-based therapies. Thank you all. By this, I have ended the class. Thank you. Thank you.